Okay, up next we have CEO and founder of D3Sciences, Nicholas Lim.
Okay, excellent. So my name is Nick Lim. I'm the CEO and co-founder of D3Sciences. I'm going to tell you today about how we'll enable conclusive and comprehensive diagnoses with the first biopsy while enhancing patient safety. I'll start by talking about the need for, if I can get the slide to advance, uh, about the need for our innovations. We've all been affected by cancer in some way or another, but most people are not aware of the fact that between 1 in 4 and 1 in 5 initial needle biopsies fails to capture enough of a tissue sample to provide a comprehensive and complete diagnosis, a conclusive diagnosis. There's nothing worse than telling a patient and her family who have been waiting for biopsy results that the results, in fact, themselves are inconclusive. Uh, the failure in the first biopsy instance can also lead to the question of, is there another biopsy prescribed, or does the provider end up giving a best guess at therapy, a broad therapy, which, as we all know, results in highly undesirable side effects. Now, there's also another untold but very serious story with regard to needle biopsies, and that's complications. The most common and also most serious of which is bleeding. Depending on the vascularity, the organ, and the skill of the provider, complications can occur. Major hemorrhage can occur between 0.1 and 8.3%. As an example, spleen ranges up to 8.3, and breast, being a fatty organ, starts at about 0.1. Major hemorrhage requires transfusions, interventions, sometimes organ removal, and in the worst of situations, the patient, in fact, dies. And having talked with many, many radiologists, I can assure you that the radiologist will remember patient deaths for the rest of their lives. Tumor seeding is also a complication that is often discussed. However, tumor seeding is less common, happening about 3.4% of procedures. As far as bad outcomes are concerned, this is truly bad outcome territory. Now, our mission is to improve tissue sampling with increased patient safety to arrive at a 100% successful diagnosis rate on the initial biopsy. Our flagship invention, the Triloxis needle biopsy device, uses vacuum suction to collect a larger tissue sample, and as an electrified device, also has the ability to stimulate electrocoagulation. With the first biopsy yielding a conclusive diagnosis, it's possible to deliver personalized therapy sooner, empower the shorter and lower cost cycle of care overall, and by this means ultimately improve patient mortality. We have two patented inventions. I'm only going to be talking about one of them today, and that is our flagship Triloxis needle biopsy device. As I mentioned, it is an electrified device, self-contained. There is no external power supply or vacuum generator. It uses vacuum suction to collect a larger tissue sample, as well as any surrounding fluids present with the suspicious tissue. It has the ability, as I just mentioned, to initiate coagulation at the excision site, and with that capability, reduces the risk of complications, as I mentioned, the bleeding, infection, and tumor seeding. Leads to, as we say here on the slide, the comprehensive and personalized diagnosis, which is typically associated with not just a test to determine the presence or absence of cancer cells, but rather beyond those tests to move to genetic and molecular testing, which of course enables personalized therapy like immunotherapy. The patient experience is better. You eliminate the need for multiple biopsies. The delivery of care occurs faster, and outcomes also improve. Now, as I mentioned, Triopsy is a electrified device. It has the ability, it is the only device that has the ability to perform a biopsy and to perform electrocoagulation all in one device. Time is money, and Triopsy makes very efficient use of facility and provider time. As far as TAM-SAM-SOM is concerned, global market for soft tissue biopsy devices is 2.5 billion, growing at about 7% per year. Of the 2.5 billion, 1.5 billion is Europe and North America. Within that 1.5 billion, about 800 million is North America alone. Triloxis is a single-use device, and we expect that the obtainable market will be just over 100 million. Here are our milestones. We've got the 3D printed models. We expect to have a functional prototype within the next 2 months. We have started our animal studies at UC Davis utilizing a coagulation probe on dogs with fatty liver disease. We have a fundraising round open now, our first round. With those proceeds, we intend to recruit staff and undertake the FDA and reimbursement work that needs to be done. The seed round we have now, it is actually a safe offering. Will take us to the point of applying for FDA clearance. It's our intention to have that be our potential first exit point. And barring that, we'll need to raise more funds to make our way through to the post-clearance time period. If we aren't acquired at that point, then we will execute our go-to-market plan. The CAP plan, I mentioned that we're starting off with a safe offering to raise $500,000 with the terms that you see right here. We're undertaking that right now. The overall seed round is targeted at $2.5 million. That, as I mentioned, will then be followed by Series A, which is intended to take us through the commercial launch preparations. The team. I've had 12 startups, out of which I've had 2 successful exits, a single and a triple. I'm quite confident that D3Sciences is going to be a home run. My co-founder, Dr. Mike Larson, is our Chief Science Officer. He's a practicing radiologist, a researcher, a prolific inventor. And as you can see on the slide, he's faculty at UC Davis Medical School, in addition to serving at the medical center as their director of cross-sectional interventional radiology. We have 2 advisory boards, one medical and one business. On our medical advisory board, we have 3 interventional radiologists, 2 mammographers, and a pathologist. Our business advisory board, our business advisors are familiar with startups, with medtech, with needle biopsy devices specifically, and of course, with raising money. I'm going to come back to this slide, but these are some frequently asked questions that we encounter. I touched on the fluid collection, what our regulatory pathway is going to look like. Why are we an attractive target for an incumbent in the market? And how do we know that successful initial biopsy reduces the cost of care? As I said, I'll come back to this slide. And at that point, welcome all of your questions. I'm sorry, that's not what I'm looking for. I apologize. Let me get to summarize our my presentation. I apologize. To summarize my presentation, providers, facilities, patients all benefit from the use of Triloxis. The market is a sizable global market available to us. We have a quick regulatory pathway, 510(k) pathway with plenty of predicates, a team that knows what it's doing and has the ability to carry this forward. And last but not least, that $500,000 we'd like to raise right now in a safe offering. So let me go back to the prior slide, the frequently asked questions, and I invite your questions now. Please ask me whatever you wish. Perhaps just to get things going, I'll use these questions. I think you can see on the slide that with fluids and tissue together in a biopsy, the efficacy of genetic and molecular testing rises to 90%. It falls quite a bit short of that in the high 60s if tissue is all that's collected. We have plenty of predicates associated with coagulation, cauterization, et cetera. And we have the animal study protocol as well for that 510(k) short pathway. The needle biopsy market is a saturated market. In order for the incumbents to generate more revenue, they have to break out of that zero-sum game trap that they're in and innovate ways by which they can expand the market. The features of Triloxis, and I only touched on a few of the features in this presentation, but the features are such that an incumbent like Becton Dickinson or Merit Medical can spread those features across their existing product lines, and with these capabilities of coagulation, vacuum suction for greater tissue collection, et cetera, expand the market. We have existing research in published peer-reviewed papers that discusses the cost and time delay effects associated with repeated biopsies on patients. Any questions from the audience? Did I impress you so much that you are ready to give money without asking any questions?
Yeah, the money was already wired. Didn't you get it? No.
Oh, that might be.
Sorry, that was the went somewhere else. Do have a question. Can you explain a little bit more about your economic model? So I understand, how you would ultimately generate revenue on this, and who is the actual customers and potentially the distribution channel?
Sure. As with many medical devices, the customer who promotes and champions the device is the user, the providers who perform biopsies. However, the customer who cuts the PO and pays the bill is the hospital. And this is where the value-based analysis becomes extremely important. We intend early on to have a direct sales model with our own market access professionals involved who will take the value benefit analysis to CMS for reimbursement code purposes and would accompany our salespeople to pitch to group purchasing organizations, and then after those GPOs to individual hospitals. The economics, again, the device is single use, and it's extremely cost effective. So I'm not going to dwell on this, but we have an interim bill of material, a prospective price for the device, and we understand what the sales cycle will be to to acquire customers. Did that answer your question?
Yes, thank you.
I should also add that once we have a presence in the marketplace, that we will recruit an indirect channel, catalogs, for instance. There is going to be a certain amount of time associated with that because our intent is to create localized markets around our medical advisory board members who are based in places like New Haven, Connecticut, Gainesville, Florida, and the Denver metro area, as well as Northern California. So with the establishment of local markets around our medical advisors, we'll be able to collect data, from those highly controlled and individual areas. And that data, of course, will be factored into our work in analyzing value benefit.
Okay. Thank you so much, Nicholas.
Thank you. Appreciate it. Thank you, all the good folks at Aquillius.